Good morning. It's my pleasure to welcome you, those of you in person and those of you online. It's great to have you with us for our late fall, we'll call this, corporate breakfast. This morning's breakfast is sponsored by the Education and Membership Committee of the Development Council. Bob McGoy serves as the overall chairman of the Development Council. Dave Shull serves as the vice chair. But thanks again for joining us. We try to have 4 of these each year. And once again, we're glad you are with us. Jeff Smith serves as the Director of Pastoral Care here at the Health Systems and will lead us in our invocation.
May we bow our heads in prayer. Dear Heavenly Father, we acknowledge your rightful place in our hearts and our lives. And we want to thank you for the many blessings you showered down upon us even during this time. Father, we want to lift up our medical staff and our nursing staff and all the ancillary staff. Father, I lift them up to you and ask that you continue to give them perseverance and fortitude and peace, Lord, and rest as we continue this fight. Father, I want to lift up the scientific community as they develop and implement a vaccination plan. Father, and I want to lift up our government, our leaders, ask that you give them wisdom and discernment as they draw us together. Father, we pray for your guidance, your wisdom, and your discernment in all that we do. Use us as your instruments, guide us with your love, and help us each and every day. In Jesus' name we pray. Amen.
Thank you, Jeff. My buddy Jim went to the dentist a couple weeks ago, and unfortunately, the dentist said he had a mammoth cavity. I mean, it was huge. In fact, it was so big, his dentist sent him to a podiatrist. Farmer Giles took his pigs to the market every Wednesday for years. Never sold an animal. Never sold an animal. Well, a couple of years after he stopped doing this, he realized his mistake. Market day was on Friday. A couple of fleas are planning a trip to the other side of the house. One flea says to the other, "Shall we hop or take the cat?" And finally, Bill is standing in the bathroom, standing on the scales, sucking his stomach in desperately. I mean, he is just almost uncomfortable with sucking his stomach in so much. Well, his wife comes in and tells him it's not going to do any good at all for you to do that. He says, "You're darn right it's going to do me good because that's the only way I can see the numbers." Rob Antagny serves as the President and CEO of the Health Systems and will introduce our speaker today.
Thank you, John. And it's good to have many of you out there virtually, some of you here socially distanced in person. Just a reminder, those of you that are watching this on our YouTube, if you do have questions, I actually have a screen, and if you type those in, I will monitor the ability and try and relay those questions to Dr. Karen. So in advance, if you already have some thoughts, don't hesitate to send those out. Before we start with the program, have an opportunity to welcome a number of new physicians to our medical staff and our community, I think. Or not. Check, technical difficulty. Our IT folks are scurrying as we speak, but we actually have been able to recruit a number of new physicians even during this challenging time. And obviously that's very important that we're able to continue to bring in talent. We'll see if we can get their pictures up here. There we go. So Brad Lawson is someone that should be familiar to us. He left for a brief time and has rejoined us. as an otolaryngologist. He earned his medical degree from The Ohio State University and completed his residency at Arkansas. So he is certified in neck and head and neck surgery. So he's joining Dr. Parker once again, and once again, many of you probably are familiar with him. Out in Johnstown, we have a family practice physician who will be joining us, Dr. Elizabeth McIntosh, MD, once again working in the Johnstown area. She earned her medical degree from the State University of New York, Upstate Medical University in Syracuse, completed her residency at Grant Medical Center in Columbus, and is board certified in family medicine. Next we have Olubenga Felix Tolani, MD. He is feverishly working hard right now as one of our hospitalists, as we've seen a bit of an uptick in our inpatient. So he's working up there on the floors, earning his medical degree from the University of Lagos College of Medicine in Lagos, Nigeria. Completed his internal medicine residency in Brooklyn and is board certified in internal medicine. And once again, if you're an inpatient, you will see him working as a hospitalist. And I think that completes the introduction of our new physicians. So without further ado, I know we have great attendance in terms of the virtual queue, if you will. I think we have roughly 60 to 80 that are on line watching this, and another 20 or so here. So a lot of interest in what I asked to be put together for today's program, and really for a number of audiences, was a nonpartisan, non-politically motivated review of all the vaccines that are in trial, what they've gone through, their efficacy potentially, and just sort of the facts, going back to the, what was that, Craig? Was he Sergeant Friday? What was his rank? Just the facts, ma'am.
I think it was
Joe Friday. Joe Friday. Come on, give me some help here, people. So anyway, we're just going with the facts today. We're sticking with what is known, and of course this evolves. Poor Craig did this a couple weeks ago, and I think he's had to update it almost daily since he completed it, but it will be truly up to the minute. A little bit about Dr. Cairns, MD, MPH. He is our Vice President of Medical Affairs. He oversees our patient safety and process improvement efforts here at the hospital, and he also serves as the liaison between the medical staff and the hospital administration. He is our hospital's chief medical officer. He also worked as a physician for the health professionals starting back in 1999. He has worked for well over 20 years. What's it, more like 40 years in the community, Craig? Probably 40 years. I think 20 years maybe employed by us, but 40 years in the community. He earned his medical degree from the Ohio State University College of Medicine. By the way, John, your jokes were okay, but I really appreciate you not bringing up Michigan's football team in any of your opening remarks. So I will do it for you so I don't get any comments through the YouTube. Craig was able to get into the Ohio State University College of Medicine, however. He also earned a public health degree from the Medical College of Wisconsin. Did his residency at Riverside Methodist, board certified in multiple areas, resides in Heath, and is the father of 3 grown sons, and he's going to give you all the facts about the vaccination process and vaccine availability.
Thank you, Rob, and thanks to all that are joining us online. And as Rob mentioned, I'll try to give you an update on the current status of the coronavirus vaccines. I think this is a very timely topic, as it turns out. Also, a little bit on the current state of coronavirus therapeutics, and just a few slides on an update on influenza immunization as well. So this first slide comes to us from the New York Times disease vaccine tracker, and they're very good about updating this. This looks like it was updated. If I can get my pointer to work here. Looks like it was updated on October 20th. I think that's when I sent my slides over. Actually, it was updated as late as yesterday, and actually the numbers there are unchanged. They're still the same. So they go through the different phases, phase 1, 2, 3. They say that there are 6 vaccines that are approved for limited use. As it turns out, all of those vaccines are in other countries. None of them are approved in the United States for limited use. I think 2 of them in the Soviet Union and the other 4 in China. So what are these phases? We have this preclinical phase, which primarily is taking place in animals. And I think they're looking to see if these vaccines are in any way immunogenic before they progress on, and also if there's any obvious toxicity that would stop the trial right there. Phase 1, you have a very small number of people, again, testing mainly for safety and looking for an effective dose that provides the immunogenic response that you need from the vaccine. Phase 2, they increase this to hundreds of people, and in this phase they segregate into groups. They look at male versus female, elderly versus young, different ethnic groups to see if there's a difference in the response. Again, also looking for safety in this group. Phase 3, where we have a number of vaccines in phase 3 right now, this is where you get into these randomized trials, and these are large, anywhere from 30,000 to 60,000 volunteers. These are randomized, double-blinded, placebo-controlled trials. So the person receiving the vaccine, the physicians or caregivers that are administering it, and the regulators, no one knows who receives the vaccine and who receives the placebo until the end of the trial. And again, we have a number of vaccines that have progressed to this level. And the FDA is looking for at least a 50% efficacy, I think, before they grant approval in phase 3. And then if it gets through all of these phases, then the FDA might grant emergency use authorization, particularly in an emergency situation like a pandemic or full approval. So these are the different types of vaccines in general, and I think if we're looking at the coronavirus vaccines, are primarily either protein-based or gene-based vaccines, and we'll talk about these different gene-based vaccines. So these are some of the examples of the vaccines that are in phase 3. These top 2 are in China, and these have been granted emergency use. Soviet Union, or Russia, I guess, and China, they're not as particular about the safety protocols that we do in this country, so their system is much accelerated. The one that's been in the news the last couple of days is the Pfizer vaccine, which is a messenger RNA type of vaccine, very similar to the Moderna. These are very similar vaccines. Both of them utilize this technology called messenger RNA. The AstraZeneca vaccine is a viral-based vaccine that was researched at Oxford University in England. And I have down here on hold, this is kind of an old slide. This was on hold because of a concern in one patient of some spinal cord inflammation, and that since has been removed. It's no longer on hold. The same way with the Johnson & Johnson vaccine, another viral vector vaccine that was on hold for an undisclosed toxicity, which again has been, the hold has been released. It seems to be a very safe vaccine. This chimp adenovirus, it is interesting. They tag the DNA of the coronavirus into another virus, usually an adenovirus, and if your body has had experience with that virus before, it might actually attack the virus and decrease the efficacy of the vaccine. So what AstraZeneca did is they used what they call a baculovirus, which is a chimpanzee-based virus for the adenovirus. The Johnson & Johnson vaccine does use a human adenovirus. So I'm sure everybody is familiar with this scheme of the virus. We have the endoplasmic reticulum, or the ribosome, where the RNA of the virus resides. As you see, these are RNA viruses, so there's no nucleus of the virus, there's no DNA. And then this is the area that is being targeted by these vaccines, the so-called spike, the spike protein or the S protein. So this, to me, this is the most exciting technology. These are the messenger RNA vaccines. This one is Moderna. Again, it's very similar to Pfizer, which is the one that's been in the news. And how this works, you look at the coronavirus, and they have known for quite a while the entire genome of that virus. So they can map out all the genes in that virus, and they not only know the genome, but they know which segments of that genome code for different proteins, the so-called codon. So they take the codon that codes for the S protein or the spike protein, they infuse it with a little lipid so that it's absorbed into the system. They inject it into the deltoid muscle. It goes through the muscle apparatus and ends up in these dendritic cells. And these dendritic cells tend to be the most immunogenic cells in our body. And from there, it activates lymphocytes, T lymphocytes, CD4 lymphocytes, as well as CD8 lymphocytes, which are known as the killer cells. And in addition to lymphocytic stimulation, it also activates the B lymphocytes and plasma cells, which make the antibody. So you get high levels of neutralizing antibody, which is the only type of antibody that counts, as well as cellular immunity. So it's a very effective mechanism. And some of the advantages here, again, this is totally, the reproduction of this is, there's no active virus involved, and there's no requirement for incubating this in eggs like we do with a lot of vaccines, particularly influenza vaccines. This engineered messenger RNA, the way that it's produced facilitates large-scale production. You can crank out a lot of this vaccine in a short period of time. You get high levels of neutralizing antibody as well as the cellular immunity with the T lymphocytes. You don't have a viral vector like you do with the AstraZeneca vaccine and the Johnson & Johnson vaccine. So you don't have this idea of pre-existing immunity. There's no antibody in our system that attacks this because it's totally novel to our system. And the other thing about the viral vector vaccines, so these are DNA-based vaccines, and for these to make the protein, the DNA has to actually make messenger RNA, which then goes on to produce the protein. And for that to happen, the DNA has to enter the nucleus of the cell, which only happens during times of cellular division, which decreases the efficiency of the vaccine. The other theoretical concern is that you're actually taking foreign DNA and mixing it with our own DNA, and some people have a little bit of an apprehension about that, thinking that it might actually alter our own DNA. And the other nice thing about these vaccines is there's no active viral material in this. These are totally reproduced or totally produced segments of RNA. They're non-replicating, so they don't regenerate themselves. And being a foreign protein, shortly after it induces immunity, the body gets rid of this DNA fragment, so it's not left behind. And as far as we know, there really isn't any potential for these vaccines to actually cause infection. The major disadvantage is the storage requirement in a sub-zero environment, anywhere from -70 to -80 degrees Celsius, which translates to which translates to about negative 112 degrees Fahrenheit. So these again are some of the vaccines that are in phase 3, both the Pfizer and Moderna, very similar vaccines. They have a little different dosage. Both target, all these vaccines actually target the spike protein. Both of these are messenger RNA vaccines. There's a little difference in the dosing schedule. Both of them require 2 doses. The Pfizer is 2 doses 21 days apart. The Moderna is 2 doses 28 days apart. There haven't been any serious side effects noted in phase 3 trials with either one of them. Again, the AstraZeneca is probably going to be a very immunogenic, very good vaccine. It was temporarily on hold because of this issue of transverse myelitis, which subsequently has been reversed. The big advantage with the Johnson & Johnson vaccine, another viral vector vaccine, is that it only requires one dose, which is good. I think particularly with having to comply on, or having to depend on people to comply with coming back and getting the second dose. And this ADE is a concern with vaccines. What this is augmented enhancement of the protein. And again, as I've mentioned, the only antibody that is relevant is a neutralizing antibody. If a vaccine makes non-neutralizing antibody, what they've found is that antibody complexes with the virus, and that complex will stimulate another receptor on our cells called the F receptor that actually facilitates active virus into the cell. So they want to make sure that these vaccines, if they're measuring antibody, if they're measuring neutralizing antibodies, so that you don't get this enhancement of non-neutralizing complexes. This is a slide from the 19th century, and it had to do with Jenner's smallpox vaccine. As you remember, what Jenner did is he used another virus that caused a lesser disease, cowpox, and we had anti-vaxxers even back then. And the context of this cartoon is that if you receive his cowpox vaccine, you indeed might develop some bovine type of features. So we see these people with horns and other bovine features, and this fellow here, I'm not real sure, he's scratching something down there and delivering a calf or something at the same time. Anti-vaxxers are not anything new. This is an interesting website from the World Health Organization. They list a lot of information as well as disinformation about not only the vaccines but the coronavirus in general. And these are just some of the examples that are on that website. There's many more. And these are some of the concerns that are prevalent on the internet. COVID-19 vaccine might be the mark of the beast, where they're referring to this autocratic individual in the biblical book of Revelation that requires his mark for people to participate in the economy, that masks themselves might cause lung disease, which they do not. And this one I haven't really seen, but I guess it's on the internet that it's a conspiracy from Bill Gates who will inject a microchip with this so that he can control your activities. So this is a quote from Cameron's book, The Plague, that stupidity has a knack of getting its way, which is certainly true today as it was then. So a few slides on therapeutics or treatment of coronavirus. And treatment is segmented, I think, into the degree of illness. So we have patients that we consider to have non-severe illness, and people can feel really sick. That doesn't mean that they aren't horribly ill with this, but this is dependent on their need for oxygen. So if their oxygen saturation is less than 94%, they're considered to not have severe illness. Severe illness, if their oxygen saturation falls to that level, or if they need supplemental oxygen, that is severe illness. And then critical illness is defined as patients that are in septic shock, or those patients that are sick enough to require ventilators, or extracorporeal membrane oxygenation, which is a sophisticated treatment if you're really, really sick. So again, this is remdesivir, which has been in the news, or Veklury is the trade name. It's made by Gilead, and it's probably the most effective therapeutic that we have right now. This is a schematic of how it works, and it works by inhibiting replication. And this is the enzyme that it inhibits, this RNA-dependent RNA polymerase. And I think when this was first released, we were real cautious here at Licking because we were afraid we weren't going to be able to get the drug, and we restricted its use. And We were using it only in the sickest patients, and we required approval of our infectious disease specialist to order it. And that was kind of the wrong thing to do. What we found is this drug is much more effective if it's given early on. Probably the earlier in disease, the better. Again, it's inhibiting replication, so as soon as you can inhibit replication, the better the patients are going to do. And one of the disadvantages of the drug is the only way it can be given right now is intravenously, which really decreases the ability to provide it on an outpatient basis. Although Gilead right now is entering into trials an inhaled version of this, which might really expand its usefulness to outpatients. It's basically a pretty safe drug. There is some concern about patients that have kidney failure. It shouldn't be used in patients with kidney failure, and also patients that have liver disease. It has to be used with caution. We measure liver enzymes as part of the administration of this drug. Another therapeutic that we have is convalescent plasma. We've used a lot of this. Again, this is an intervention that is much more effective if it's given early on in the disease. This is an old technique. In fact, I know when I came to town 40-some years ago, there was an older pediatrician who retired, John Fleet Miller, and my partner was his doctor. I know I made house calls, and he was a real gentleman. And I had another elderly patient who had polio as a child, and he told me that when Dr. Miller had a child with polio, he'd come to him and take his plasma to give to the child with polio, and it was an effective intervention, even back then. But this was something that's been used for a lot of infections. The idea is that if someone has been infected and they recover and they have antibodies in their system, those antibodies might help somebody else, as well. This really isn't approved by any regulatory body, but it's pretty widely used. I think everybody is using it. And the other thing is you really don't know how much antibody is in the units that you receive. So in general, we usually give 2 units at a time just to make sure that people are getting enough of the antibody. Now, both remdesivir and immune plasma have to be given early in the disease. Dexamethasone, which is a cortisone type of medicine, is different. This is one that's much more effective if it's given later in the disease. And what some people get is an elevated immune response, and the immune response kind of overacts what they call cytokine storm, and attacks the lung tissue and blood vessel tissues, and can be very harmful to the body. And dexamethasone kind of wants that response. I think back with the 1918 influenza, that was the main killer. That was a very unique infection in that it was much more deadly in young, healthy people, and that was why the people that were able to mount an immune response, again, they got into the cytokine storm, and the antibodies would wipe out the respiratory epithelium, and then they get secondary infections with pneumonias with things like staph, which they didn't have any treatment for, back then. Again, this is approved by the ISDA for severe and critical illness, patients in later stage. It's not really approved for non-severe illness, early on. This is one that's been in the news. This is what President Trump received, this polyclonal antibody. And the reason that it is polyclonal is they take 2 monoclonal antibodies and fuse them together. One is a human derivation, the other is of mouse or murine derivation. And the way that this works is that this antibody attaches to this spike of the virus and inhibits its entrance or attachment to the ACE receptors, and so it doesn't get into our cells. Seems to be a very effective treatment. It is not yet approved for use. I know they've applied for emergency use authorization, And Lilly has come out with a monoclonal antibody. I think it was just approved for emergency authorization yesterday, called bamlanivimab. And that was Sony, I didn't include that in the slide, but it kind of works the same way. But instead of having two monoclonal antibodies fused, it's just a single monoclonal antibody. These are more theoretical drugs. Again, this is to inhibit this cytokine storm. These are drugs that are already approved for other things, mainly for autoimmune disease and cancers. And they can be used also, I think, if people are getting into trouble late in the disease with the cytokine storm. Famotidine is the Pepcid, which is an over-the-counter medication. I'm not real sure how that works, but it does seem to be somewhat helpful, decreasing both the mortality rate as well as the time to symptom resolution. So again, it seems to be a very safe therapy that is somewhat effective. And then anticoagulants are really pretty much recommended for all inpatients. For some reason, this virus attacks blood vessels, both arteries and veins, the lining of the arteries and veins, and induces clotting. So you end up with arterial occlusions. We see young people that are affected with coronavirus that end up with heart attacks and stroke, as well as people that are developing phlebitis. So I think all of our COVID patients we put on, we usually use low molecular weight heparin here in the hospital, and then there's some concern that maybe even after they go home, they ought to take an oral anticoagulant for up to 45 days or so. Okay, and just a few words here on the current status of this year's influenza vaccines. There are many options available. We have vaccines that contain 3 viruses, some that contain 4. We have high-dose vaccines for people in my age group. We have vaccines that have adjuvants. We have recombinant vaccines that don't have any viral material or that aren't grown in eggs. Cell-based vaccines that are grown in other mammalian cells rather than hen's eggs. For people that are afraid of shots, we have nasal spray vaccines, as well as a vaccine that can be administered via injector. So these are the egg-based vaccines that we have this year. These are the 3 viruses in the trivalent-based vaccines. And it's the same 3 viruses in the quadrivalent, plus they put another B virus, a Yamagata-linkage virus, to have two A's and 2 B's in that. This is the intranasal. This is usually used in kids, but it can be used in adults up to age 49. So it's approved for ages 2 through 49. It's recommended that all kids over the age of 6 months be vaccinated unless there's a contraindication. So if they're younger than 2 years and older than 6 months, they have to get a shot. But if they're 2 years to 9 years or older, they can get the nasal spray. The dose is a little bit different depending on the age. I think in the younger children, it requires 2 doses. If they're over 9, they can get by with 1 dose, unless they had received 2 doses the year before, and then they can get by with one dose. It has a little different viral composition than the quadrivalent egg-based vaccine. It's not to be used certainly if people have a prior allergic reaction. Usually live viruses, it's not a good idea to give it to pregnant women. And it's they say to use it with caution in patients with asthma, since it could inflame the respiratory system. It is a live virus, so again in kids, if they're on aspirin therapy, we always worry about this Rise syndrome, so there's a caution there as well. These are egg-free vaccines, recombinant vaccines, or flu block. These are made synthetically, kind of along the same line as the messenger RNA, although these are DNA-based vaccines. And again, it's like the AstraZeneca, they combine it with a non-human virus like the chimpanzee virus that AstraZeneca uses. We have cell-based vaccines, which are again, recombinant, and these are grown in mammalian cells rather than eggs for people that are allergic to eggs. Again, a little different viral composition, but 2 A's and 2 B's, which is what seems to count. These are new. They're new mainly because this year they're quadrivalent. They had been trivalent in the past. And there's 2 options. The one we have here at Licking is the Fluzone, Sanofi Pasteur vaccine. These are egg-based vaccines. The other option is one that uses an adjuvant. And the adjuvant they use is called squalene. That is an oil that's present in a lot of places, particularly in fish and really prevalent in shark liver. And it's used not only in vaccines, but it's also used in cosmetics as a moisturizing agent. I hear there is kind of an anti-squalene movement to save the sharks and their livers from being used in this manner. But these are highly effective vaccines. Again, if you're over 65, these have 4 times the amount of antigen of the regular quadrivalent vaccine, so it's recommended in that age group. The jet-based vaccine, which is Afluria, again, this avoids a needle. Maybe one disadvantage with this is it does contain thimerosal. There's still a lot of people that are very reluctant to receive vaccines that have thimerosal, although there really hasn't been any evidence that it's toxic. I think the type of mercury that's used in this is the ethyl mercury. It's the methyl mercury that causes neurotoxicity. But at any rate, these are vaccines that contain thimerosal, and as you can see, that the amount of thimerosal is minuscule in all of these vaccines. So who should get flu vaccine? And flu is still important even with coronavirus going on. So it's recommended in everybody over 6 months of age unless there's a specific contraindication. It's particularly relevant in kids. They tend to, as opposed to coronavirus where they do well, with flu they can get really sick, and it's much more toxic a lot of times in children than it is in adults. Certainly older adults are more prone, and for some reason flu is very serious in pregnant women, so we really recommend immunizing all women in their pregnancy, and certainly if there are underlying conditions. This is some of the data that everybody's familiar with. Can cause 30,000, 40,000 deaths every year with influenza. We have a lot of hospitalizations, a lot of illness and morbidity that's caused by the flu virus. They're predicting this year it's not maybe going to be as severe. I think we look at the southern hemisphere. Flu usually occurs in the summer. Their seasons are reversed. Their summer is our winter and vice versa. And in Australia, it was a fairly mild flu season. And they're thinking that with all of the precautions we're taking with coronavirus, with masking and social distancing and sanitizing, that is going to be helpful in preventing influenza, as well. People should be getting their flu shots. It's recommended by the end of October, although it's still, if you haven't got your flu shot, they're still available. We recommend that you get them. Some of the concerns with COVID, we don't want people standing in long lines if they have COVID, obviously, to get a flu shot and infecting everybody else in the line. Wait until you're through your quarantine period and then get your flu shot. Again, if you're hospitalized and ill with COVID, wait until you're better to get your flu shot. And if you have moderate or severe illness with COVID, you probably should wait until you're no longer acutely ill to get the shot. So these are a couple of quotes I thought I would throw in. And George Bernard Shaw says, "We learn from history that men never learn anything from history," and I think he was prescient. I think that's certainly true today. And I like this other quote here from Ken Rule. He said, "Simply say, we learn in the midst of plagues, there are more things to admire in men than to despise." And I think in this political climate where we hear so much negative stuff about infection immunization and everything that has to do with epidemic control. And I look here at Licking, and I think we have a lot of, not just men, but certainly men and women here to be admired. I think our administration has provided with protective equipment and resources to be adequately to protect not only our staff, but our patients, certainly the caregivers, the physicians and nurses and other clinical people on the front lines, as well as all the other staff, you know, the housekeepers that sanitize the rooms, you know, everybody that does clerical work. There's just a whole lot to be admired. So with that, I'll end and take any questions that you have.
Well, thank you, Craig, for a great presentation. We have a couple of questions, but you left yourself out, and men to be admired or people to be admired. Certainly, Craig and our infection control folks have really led a tremendous effort in this endeavor. So I did get a question, and I think it's a good clarifying question. The COVID vaccines, are any of them live, and what are the specific benefits, in your opinion, of the messenger RNA viruses versus a live vaccine?
Well, again, the messenger RNA is totally synthetic. You're taking this segment of RNA that is made synthetically. There's no active virus. It's non-replicating, so it doesn't have the ability to reproduce itself, and the body destroys that segment shortly after it induces immunogenicity. The viral vector vaccines use the DNA of the coronavirus, but it is used in a live virus to get into the system and induce the immune response. And the two main viruses in that category are the AstraZeneca vaccine and then the Johnson & Johnson vaccine. Again, the AstraZeneca vaccine uses a non-human adenovirus from chimpanzees. The Johnson & Johnson vaccine uses an adenovirus that has been used in many other vaccines and is proven to be safe. These are both DNA-based vaccines, so again, they have to get into our cells to make the messenger RNA to make the antibody to the protein, whereas the messenger RNA vaccines totally bypass that DNA step. They don't have to get into the nucleus, and they basically are able to make the protein without DNA.
The two closest, just to clarify, the Pfizer and the Moderna are not live vaccine.
That's true, yes.
Just want to clarify that for the question. So it is not live like perhaps other vaccines in our world. Related to the flu vaccine, we got a question. Why is the quadrivalent recommended only for those 65 and older? Why not for other populations?
Oh, no, there's quadrivalent for everybody.
Okay.
The only high-dose vaccines that are available now are quadrivalent, so you get an extra V virus included in that.
So all folks getting the flu vaccine would be getting quadrivalent?
Here, that's all we use is quadrivalent. They still make trivalent. I don't know why people would want to get that, because you have added protection with the quadrivalent vaccines.
Well, I like that promotion for us. So we're only using the quadrivalent here, even though they do make the trivalent. So the folks getting their flu vaccine here, 65 and older, 65 and younger, will be getting the quadrivalent. So appreciate that question from Tom Mullady. Any questions in our audience here for any folks? Tom.
Doctor, can you tell me, you were listing off the various companies and the various types of vaccines. Was there any coordination by the companies so that, hey, you do this type and we'll do that type and see who can find this cure, so to speak, faster? Or is it still a competitive capitalistic approach to things?
Let me repeat the question, Craig, for the audience. So the question was there any collaboration between the various companies working on vaccines, or was it a competitive market where all the companies were trying to get their product to market first?
Well, a lot of the reason that one of the reasons, I guess, that we've had this warp speed process is that these vaccines, a lot of them were experimented with other infections, particularly the other SARS virus. So there was a lot of work that was already done and kind of in place that everybody could kind of piggyback on. safety of the vaccines is overseen by the Data Safety Monitoring Board. And as it turns out, all of the vaccines are overseen by that board with the exception of the Pfizer vaccine. And the reason Pfizer is not is because they did not receive any funding from the federal government. So they're not really beholden to the government. They can have their own monitoring board. And all of these, of course, are overseen by the National Institute of Allergy and Infectious Diseases, Dr. Fauci's group, that has to give final approval. So it's not that Pfizer is undercutting, but yeah, I think they're going to have a competitive advantage. It sounds like they're going to be probably the first vaccine that is released, and they did have an agreement with the federal government to buy millions of doses of the vaccine when it is released. So there certainly is competitive activity, but there's also some collaboration.
Great question. The other part of that, Tom, that I think makes sense, and I think we've all heard this in some fashion, is the government has funded a lot of the production of these vaccines in advance of their approval, so at least minimizing some of that lag time between getting that approval and then having to create the vaccine. So I think many of you watched the 60 Minutes segment on Sunday, and I know, Craig, I asked him to watch it, and they're already talking about how they're going to distribute this because it's already been made and that investment was put up front. So, Chris?
Are all of the therapeutics that you went over, are they available here locally?
So the question is, are all the therapeutics that Craig discussed in terms of the treatment for COVID, are those available here locally at Licking Memorial?
Well, the remdesivir and the convalescent plasma, we use a lot of that. Certainly famotidine, Pepsid is available. Dexamethasone we use a lot, again, in late-stage disease. Actually, the monoclonal antibody that was released, we just approved, the Lilly vaccine, is actually not approved for in-hospital use. It's only approved for patients with mild disease. It doesn't seem to be effective. The way that it works, again, is by blocking entrance into the human receptors. And we don't have that yet, and maybe we won't get it because you can't use it for inpatients anyway. The other polyclonal vaccine, the Regeneron vaccine, has not been released for emergency use yet. That appears to be a useful vaccine, and I'm sure, not a vaccine, but therapeutic that I would guess we would probably get if that's available.
Yeah, I would say other than Regeneron, Chris, and some of the other smaller ones, we were able to do most all of that here locally. And really, working with some of the Columbus hospitals has made that available, which is great that we can work with them to be able to get adequate amounts of the convalescent plasma. But yes, most of that can be done right here. Paul?
Rob, do we have the ability to store the vaccine at negative 70 degrees Celsius, and if so, at what capacity?
So the question is, do we have the ability to store the vaccine at negative 70 degrees, and if so, at what capacity? The short answer is yes. We already have that capability, and we believe in an adequate amount to accommodate the vaccine that we would need, certainly, to deal with vaccinating our employees. Right now, as it stands locally, we are what's called a closed pod. I'm looking up at Ben. He can shake his head up or down. But we would be charged with vaccinating our employees as one of the high-risk, high-priority groups. Right now, the open pod, as they call it, the health department would be handling that. We've offered to work with them and assist them, if appropriate and necessary, in facilitating that if it would come around. So for what we are expected to deliver, we have adequate storage for that. So make sure, Ben, get a thumbs up from the back. So we should have that available for our folks. I don't know how available it will be in the community. I don't know how many community members will want it. There have been studies that suggest it's 50%, even on the high side, of folks who really want to get the vaccine. So the supply may be more plentiful than we expect, depending on the response. Now that Craig just mentioned that Pfizer didn't have to go through the government, those who are skeptical of the government may be more apt to get Pfizer or less apt to get Moderna. Or one of the things we have asked is that we try and get one vaccine, at least for our employee population, so our infection control managers aren't trying to track multiple vaccines with different employees to figure out maybe who's having greater success or not, did they get this vaccine? So we'll have to see if that can happen. So, any other questions? Oh, there's a few more that have come through. Okay, any projections on when a vaccine could be widely available? Any thoughts?
Well, it sounds, again, Pfizer seems to be the leading candidate right now, and they have essentially 50 million doses that should be available by the end of this year. That would be enough to immunize 25 million people, and they're projecting 1.3 billion doses next year in 2021. If you watch the 60 Minutes segment on Sunday night, the military is going to be very involved in distribution of the vaccine, and according to General Perna, who's in charge of this, it sounds like they have things pretty much set. McKesson, I think, is making the kits. And you think about the vaccine, you also need to have the bottles to put the vaccine in. You need to have the needles and alcohol swabs to administer it. You need to be able to have contracts for distribution, and I guess they do with FedEx, right now. So, yeah, they seem to be pretty well organized.
I think we'll be available. A couple more questions have come in. Our good friend Gus Andrews out in the Hanover area. What has been the typical admission rate? Is there any concern we might not be able to keep up? And the question is, actually, Craig was interviewed yesterday by, I believe, Channel 4, perhaps, asking about this. There is a surge. We have about 25 inpatients currently, which is slightly above the highest amount we had probably a couple months ago. And we are seeing more positives certainly in the last week or two. I would suggest to you we are capable of handling this. Our staff are doing a great job. We are working collectively with other Central Ohio hospitals, and I think that collaboration really is working well. To be honest, I think more so than any of the physical locations, the space, the beds, the PPE, it's making sure we maintain our staff's health. If and when they get sick or get COVID or have to be quarantined, obviously that takes them out of commission for 8, 10, 14 days. And certainly that could put a strain on our ability or any hospital's ability if the staff adequacy is at risk. But right now we continue to be in good shape. Our staff are doing very, very well. And I really can't, a question I get is, well, what do you think happened 3 weeks ago, roughly, for this uptick? Could it be some more testing? Certainly, I think we're doing more testing both as a state and locally. Certainly, I don't know if there are a lot of Halloween get-togethers, starting back up with college football and get-togethers that go with that in this area. But we definitely are seeing an uptick in and around the Central Ohio area, not just certainly Lincoln County, but we are managing it well. Let's see here. One more question, I guess. We've got a couple of thank yous. I appreciate that, Dr. Tom Hall and Chris Weisman. Veronica is not recognizing my face as a face ID, so I can't read the question that was sent to you. Technology. What are we going to do when we're... All right, so can you talk about the timing of distribution and priorities of distribution with the vaccine? And I think we touched on that a little bit. There are sort of two processes within the communities, the closed pod and the open pod. The closed pod being those identified in the highest priority, with healthcare workers being high on that list. I'm not aware that other high priority groups are in their own pods, if you will, or even if those have been identified yet, but there is a group that meets regularly on that distribution, and we can provide updates. But Ben, anything you want to add? Ben Burrows, our Vice President of sort of all things outpatient, and I'll just have to repeat what you say, so give me succinct answers, please.
Yeah, no, we are actively working with local and state officials on vaccine distribution planning. So to Rob's point, we're a closed pod, but we do have the ability
So right now, the priority groups, the answer there, going out to Sarah, is healthcare workers, congregate care facilities, so certainly nursing homes, and correctional facilities because of congregate care. I could see that potentially being extended or expanded, not that I know this, but if you think about any of the colleges and universities with dormitories, those are also obviously at-risk areas for potential outbreaks, so that's how they prioritized at this point. Once again, depending on the availability in terms of amounts, those groups could be expanded, extended, whatever the case may be. But I do think, as Craig said, most folks believe that by the end of this year, there will be some availability, and certainly early into next year. So I think that is on the horizon. Last question, I think, for anything that I see. Is the current uptick in COVID cases a seasonal phenomenon? Is the general public just being less compliant? If seasonal, when might we expect the trend to begin to decline? Once again, we're very fortunate that we have a lot of different testing mechanisms here. And what I mean by that is we have antigen testing, which gives us a very high degree of certainty, but we're able to back that up with the PCR rapid testing, which is even more accurate. And many of those tests will actually discern if it's COVID or influenza A or influenza B or rhinovirus. So we really get a sense as to what types of viruses are out there. And I think we've seen some, air quotes, regular flu, but I don't think the uptick is necessarily the regular flu being mistaken for COVID. We are seeing more COVID cases. I don't know the lack of compliance, if it's as simple as folks are getting tired of masking as often as they should, not being in gatherings. Certainly we've seen cases of folks gathering for weddings, funerals, parties, baptisms. What was the baby, what do you call it, when you, baby gender, reveal for parties. But I do think people have sort of got to a point where they're getting more comfortable in these gatherings, and we're seeing some of that lead to this spread. So I don't know that we have one particular thing that we can put our finger on. Certainly, if you go back to when schools opened up, I think there was some concern or hesitancy. At least the timing associated with those opening hasn't translated right into these increased cases. It's really been the last 3 or 4 weeks, certainly in this area. So we'll just have to be diligent as a society, keep wearing your masks, keep socially distancing, washing your hands, and really try and minimize those gatherings, especially, we're in November. So I understand Thanksgiving is just a few weeks away, and folks are going to want to get together with families, and shortly after that, you're going to be into the season of Hanukkah and Christmas and all of the other holiday New Year's Eve parties. And there will be a lot of temptation to say, well, we'll just do this one event. But those events can lead to the spread, and I think we just have to be very vigilant in how we proceed. So I don't think there are any more questions. We've got a couple more thank yous, and certainly, Craig, I want to thank you for all your work that you have done. If you don't know, Craig calls every positive that we get in this organization. Craig personally calls them to let them know of their positive tests and answer any questions, and I think that is remarkable. I think he was welcomed yesterday morning with 31 positives or something in that neighborhood. So starting out your Monday morning with calling 31 people before your day even starts, that's on your agenda. And he does it with a smile on his face and answers their questions, and we're just so fortunate to have him here. I will let John finish up. He wants to conclude the program, and I want to thank everybody for being here in person, the handful that are, and certainly all those. If any of you had any technical difficulties, please either call or email the development office, and we'll try and get you either the slides or some of the information. I know it's never ideal with the potential for some technical issues during these presentations, but a lot of compliments, so I think a lot of folks did enjoy this. So, John? We have a virtual round of applause for Dr. Cairns.
Thank you, Dr. Cairns, thank you, Rob. I just want to promote our next corporate breakfast, which will be February the 9th of 2021. And Rob will be giving his annual review of 2020, and he may need a lot of time. But we will always look forward to that each year. It's February 9th, 2021. Stay safe, enjoy the nice weather. Thank you.